My name is Stephen Gottschalk, and I would like to welcome you all to the Science of Childhood Cancer lecture series of St. Jude Children's Research Hospital. So with that, I would like to introduce Dr. Singh. I'm delighted that she agreed to present to us today, giving a lecture entitled Therapeutic Targeting of Self-Renewal of Medulloblastoma Stem Cells, Strategies for Blocking Recurrences. Dr. Singh received her MD PhD from McMaster's Medical School in Hamilton, Canada, and from the University of Toronto. And as a PhD student, she, in the lab of Peter Dirks, made seminal discovery, discovering human brain tumor cancer stem cells in a broad range of brain tumors, which really has formed the basis of her research career over the last 20 years. She is currently a professor of surgery and biochemistry and chief pediatric surgeon at McMaster's Children's Hospital and the division head of neurosurgery at Hamilton Health Sciences. And she is a scientist appointed to the Stem Cell Cancer and Cancer Research Institute at McMaster University. She also holds the Tier 1 Senior Canada Research Chair in Human Brain Tumor Cancer Stem Cell Biology and is director of the McMaster Surgeon Scientist Program. Her research is focused on understanding brain tumor initiating cells, and her lab is focused on understanding molecular mechanism which govern brain tumor initiating cell self-renewal. And she's focusing on signaling pathways in glioblastoma, brain metastasis, and childhood medulloblastoma, with the ultimate goal of selectively targeting brain tumor initiating cells. She has been very, very productive, publishing more than 100 manuscripts. She is very, very well funded with more than 14 active research grants, and her work really has made a significant contribution to the field. She really has become a thought leader in the field of brain tumor initiating cells and brain tumor biology. And without further ado, I'm really delighted that she decided to accept our invitation to present to us today.
Thank you so much, Stephen, for that far too kind introduction. It's my great pleasure to speak to you today at St. Jude's. I'm speaking to you today from McMaster University, from my office. And I'm really happy to speak to you today specifically about our medulloblastoma research program. As Stephen mentioned in the introduction, we're very focused on developing better targeted therapies for this population of pediatric cancer patients. And I'll speak to you today, I'll give you an overview of several of the exciting programs that are going on in our medulloblastoma research right now. I'll share with you some of our published work just to give you an idea of what we focus on, and then I'll be sharing some exciting unpublished data with you today as well. I'll start right now, and as Stephen mentioned, I'll pause to ask for questions in between, just in case, in between the sections of the talk, just in case people have to leave. Here we go, these are my disclosures. And I'll give you just a very small amount of focused background on medulloblastoma. I'm well aware that you have many leaders in the field who have probably spoken extensively to all of you about medulloblastoma in the past. My colleagues, Dr. Northcott and Dr. Roussel, have been leaders in this field. But of course, for those of you with your extensive clinical expertise, you know that medulloblastoma is the most common malignant childhood brain tumor. And over the course of my neurosurgery residency training, really just over the course of that and the beginning of my career, so over the past 2 decades, I've actually witnessed myself the increase in the 5-year survivorship from 50% to almost 80%, just through the sheer improvement of microsurgical techniques, through the improvement of chemotherapeutic protocols, and overall refinement of our protocols of standard chemoradiotherapy has led to this remarkable improvement. And really what we're left with is the 20 to 30% of children who fail our excellent standard of care. So the two foci of my lab's research program are to really try to better understand the children who relapse post standard of care therapy. Because unfortunately, since we seem to throw everything but the kitchen sink at these children who tolerate therapy so well, when they actually do relapse, we have very, very little therapeutic option for them. And on top of that, we have very little in terms of targeted therapies, which brings me to the second reason for focused study. And that is the fact that even in the children who survive medulloblastoma and all of the systemic therapy, we know that these children still have severe neurocognitive, developmental, and psychological side effects that are no doubt borne by the very toxic systemic chemoradiotherapies we have to give. And so, you know, still being able to de-escalate these therapies or replace these therapies with more targeted focused therapies is another goal of our program. And so with that in mind, we're focusing on the 20 to 30% of children who still relapse and recur, and in developing better models of recurrent disease, but also considering the successfully treated survivors and how we can reduce the burden of their systemic disease. And really, there's been a lot of incredibly thoughtful and beautiful work done on trying to understand medulloblastoma recurrence. And since my lab studies in parallel, both medulloblastoma and glioblastoma, and I find there's so much crosstalk between all of the three programs running in my lab, it's hard not to compare. And, you know, I have much more hope actually for our understanding of medulloblastoma recurrence, where I feel that therapy is driving the cellular heterogeneity to a bottleneck. And hopefully I'll share some data with you that also shows that. But the existing literature supports that concept of clonal evolution through therapy, and that there might be dominant clones that drive recurrence that actually may be more convergent, as opposed to the glioblastoma program, where the more I study glioblastoma, the more I'm concerned that there's actually a divergence, and that therapeutic bottleneck actually drives further heterogeneity and mutational burden that may be generating more heterogeneity and will become ultimately more difficult to treat. But with medulloblastoma, there's a suggestion that the relapse or the metastatic disease are actually so driven to a common bottleneck that perhaps they're more common to each other than they are to their cognate primary tumor. And this provides some hope that we can develop some really durable therapeutic strategies. And of course, everyone is very familiar with the molecular stratification of medulloblastoma. The 2018 census had determined, the consensus statement included that there's 13 molecular subtypes of medulloblastoma, but other work also refers to the group 3 and group 4 subtypes, which are the most aggressive and treatment refractory, that perhaps those two subtypes actually comprise 8 subgroups that actually form a spectrum of disease. Now, when you look at the 4 well-known classes, molecular classes of medulloblastoma, we have the Wnt signaling pathway driving group 1. We have the sonic hedgehog signaling pathway driving group 2. And you'll notice that both of those are known developmental, very important developmental pathways that regulate normal tissue specification. And of course, knowing a dominant pathway that governs a subtype of a tumor is very helpful in terms of developing therapeutic strategies. So the Wnt and sonic hedgehog subtypes are definitely the best studied and the most well categorized. But group 3 and group 4 have always suffered for a lack of better understanding of molecular mediators, and that's where my lab tends to focus the majority of our work. Typically, we have always studied and modeled group 3 medulloblastoma, but now we understand that there may be quite common molecular drivers between group 3 and group 4 with that spectrum of 8 subclasses. And really, we're focusing our efforts there. And of course, we think that a cell type that, in terms of the cellular heterogeneity of brain cancers, we do have a very good understanding that cells that tend to have a cancer stem cell phenotype have many mechanisms, are endowed with great mechanisms to evade therapy. Of course, they express ABC transporters on their cell surface to efflux out chemotherapy. They have enhanced DNA repair mechanisms that allow them to become more radioresistant. And in multiple brain cancers, we have been able to show that cells with a cancer stem cell phenotype are definitely chemoresistant and radioresistant. And so in fact, if these cells are the ones that seem to be escaping therapy and driving recurrence, it makes sense to be developing targeted therapies against these particular cells. And so one of the main goals of our medulloblastoma program is to try to understand the molecular underpinnings of self-renewal, which is the mechanism, of course, of stem cell propagation that is very specific only to stem cell cellular compartments. So we feel that if we can really understand molecular mechanisms of self-renewal, we can develop tractable therapeutic strategies. And so to that end, I'll bring you again, I've tried to sort of explain to you really why our focus is on medulloblastoma recurrence, but I'll pause here and then tell you as a neurosurgeon that we face significant challenges in modeling medulloblastoma recurrence because, again, a big limitation is restricted access to tissue. So typically, we haven't been biopsying children at recurrence. When they do complete their standard of care therapy, they will tend to relapse with the sonic hedgehog-driven subtype. Often the relapses are local. With the group 3 and 4 more common recurrences, we find that these are often disseminated throughout the cerebrospinal fluid and throughout, or perhaps hematogenously, but either way, spread over the leptomeninges of the brain and spine. And of course, this disease is not possible to treat surgically. The rationale for rebiopsying that disease is still a bit weak because we, at the moment, don't have a lot of clear second-line targeted therapies to offer, or rather differential therapies that could be suggested by a biopsy. Nevertheless, our community is moving towards pushing for biopsies at recurrence so that we can really truly understand the drivers of disease. But capturing the biology of recurrence has been a problem because we simply don't have a large number of tissue samples of medulloblastoma at recurrence. And that pushed my lab towards trying to understand, since cancer is a dynamic disease and we're able to model clonal evolution of the disease through therapy, can we really build a very good model of human medulloblastoma recurrence? And so this is the work of my now graduated PhD student who's moved on to medical school at University of Toronto, David Bakshinyan. So David began his PhD in my lab, very, very devoted to setting up an excellent animal model, building on our known gold standard stem cell patient-derived xenograft assays. So what we will do is take a primary untreated biopsy specimen from a medulloblastoma patient. We will culture the cells in serum-free media to enrich for the stem cell populations. And then we will engraft these intracerebellar into the same native location where medulloblastomas occur into the mouse brain. And then what we've done is we've adapted the ACNS 0395 protocol for high-risk medulloblastoma patients that we use to treat our own patients in the Children's Hospital. We've adapted that for the metabolism of immunocompromised mice. So here you see our NSG mice will receive 2 gray of craniospinal irradiation, followed by vinblastine, cisplatin, and cyclophosphamide. And so, sorry, vincristine. And so what we'll see is that these mice, from this adapted standard of care, they will garner a survival advantage. And the survival advantage is approximately 2 or 3 weeks, which may mimic a 2-year period that will elapse before recurrence in a child. And so you see here that what we've been able to build is a model that really does recapitulate what happens in our human patients. And here you see some MRI images accompanied by histology documenting the tumor relapse in both the untreated on the top and the untreated mice and the treated mice here. So with this model well optimized, we're now poised to be able to study all aspects of this model. And as you can see, our goal was to model the clonal evolution of cell populations through time. So we're able to actually sacrifice the mice at each time point and try to do some deep integrative multiomics profiling to really understand the drivers of recurrence. So here you can see that when we treat patient-derived xenografts, we see the same pattern of treatment responses we've observed in patients. And here you see 2 well-known medulloblastoma cell lines, D425 and HDMB03, And we see that when these are treated in vivo, that we do see an enhanced survival advantage in those mice. And very interestingly, because we're able to tag the cells with GFP, we're able to recover the cells from the craniospinal axis of the mice. And in fact, this enables us to study medulloblastoma in a regional or compartmentalized way, spatially speaking as well. So we can recover the leptomeningeal spread that you can see here around the brain and separate it from the leptomeningeal metastatic disease that we can recover from the spine, simply by harvesting the brains and spines separately and recovering the GFP tagged cells by flow cytometry. And here, this provides us a very beautiful window into the relapse disease, compartmentalized into brain and spine, that we can rarely capture from our human patients. And we do want to really ascertain that these cells that we've recovered from our mouse model, are they really treatment resistant? And here you can see that when we subject those cells to further treatment, these cells genuinely really are more resistant to the therapies that we've already administered. And that's a very important thing to ascertain to make sure that our experimental model is valid. Here is a very intriguing phenomenon that makes us think even more so that the recurrent disease is really driven by stem-like cells. So here you see we've subjected our cells from all of the different time stages of this model. We've subjected the cells to our typical stem cell assays. And so here you see the proliferation assays, and here you see sphere formation or self-renewal assays. This is typically an in vitro limiting dilution analysis, which is the best way to determine the frequency of stem cells in any given heterogeneous cell population. And so what you see here, the 5 colors represent engraftment. This is the control mouse here. And then you'll see radiation treated. So this is the cells harvested from the mice after the delivery of craniospinal irradiation, but before chemotherapy. Here you see RC, which represents the stage where we harvest the brains and spinal cords after the delivery of both radiation and chemotherapy. And then this is the mice at endpoint, which we've termed relapse. And so here you can see that throughout time, the cells from the different stages of the model show increasingly aggressive proliferation ability and more worrisome increasing self-renewal capacity, to the point where when we start at the very beginning of the protocol, you see a stem cell frequency of 1 in 237 cells. By the time you get to relapse, this is dropped to 1 in 37 cells. So what this is telling you is that the relapse compared, all of these profiles are done on the same number of cells harvested from each tumor. And so what we can recognize is that these stem cells are evading therapy, and in fact, they're really regrouping and dominating at relapse. And so this brings us to the point of really trying to understand. We've now shown you that self-renewal ability is greatly enhanced at relapse. And we really do want to understand what are the molecular underpinnings of this self-renewal mechanism that seems to be allowing cells to evade therapy and then persist and drive recurrence. And one of the genes that we've studied heavily in my lab, and one that has been studied by many of my colleagues, is BMI1. And as many of you may know, BMI1 is a master regulatory self-renewal gene that plays a very important role in development, and in both in normal development in normal stem cell populations of both the blood and the brain and many other organs, as well as their neoplastic or cancer stem cell counterparts. And here you can see that my colleague Sylvia Marino really showed beautifully how BMI1 is essential for its cerebellar development. Indeed, the knockout mouse for BMI1 tends to have really badly developed cerebellum, as well as anemia. And you can see that Sally Temple has also profiled a very important role of BMI1 in normal neural stem cell self-renewal. My colleague Jeremy Rich has already moved on to targeting glioma stem cells through different combinations of polycomb group inhibitors. We've been working on BMI1 for some time now, and in this paper from many years ago now, we were able to show the interplay between the sonic hedgehog pathway and the cross-regulation of certain mediators of sonic hedgehog, such as the GLEs, in cross-regulating BMI1. Differentially, depending on whether you're a stem-like CD133 positive cell versus a non-stem-like CD133 negative cell. And we further went on to recapitulate some of the very elegant work that Sally Temple did in normal neural stem cell populations to show that FOXG1 BMI1 axis that is present and regulates normal neural stem cell self-renewal also is dysregulated and plays a role in driving tumorigenicity of medulloblastoma stem And so here I'd like to bring you to the point to really let you know that BMI1 not only is a regulator of cancer stem cell self-renewal, but as many of you know, it's also an epigenetic regulator. And epigenetic regulators have played a huge role in medulloblastoma, as medulloblastoma is known to be a disease with dysregulation of DNA methylation, histone modification, and other ATP-dependent chromatin remodelers. And we think that the mutations and copy number alterations and chromosomal gains and losses we see in medulloblastoma occur in a large extent to these epigenetic regulators. And this is especially the case in group 3 and group 4, those two more aggressive subtypes of medulloblastoma, in which some tumors are reported to have a very low mutational burden and perhaps no mutations at all, but they have very high levels of H3K27 trimethylation. And this epigenetic mark results in gene silencing by chromatin condensation at promoter sites of fate determination genes. So we know that chromatin remodeling is regulated by this complex and highly dependent interplay of multiple genes in the polycomb group complex, and BMI1 belongs to the PRC1 complex. And there's an interaction with the trithorax gene complex family, which have an antagonistic effect on the polycomb group complexes. And so we think that in medulloblastoma, there's an imbalance of these epigenetic regulatory mechanisms that results in inappropriate silencing of genes that normally cause differentiation. And instead, we see that genes that are driving self-renewal are de-repressed. So interestingly, the epigenetic regulation of fate determination and self-renewal that we think is undertaken by BMI1, we see BMI1 is highly expressed across all medulloblastoma subgroups, but especially in groups 3 and 4. And you can see at the side here that high BMI1 expression is associated with very poor survivorship, not only in medulloblastoma, but in a study done by Glinsky et al. Across 11 different types of cancer. So BMI1 is definitely a predictor of poor survivorship. And we know that as BMI1, as shown here, functions as part of the PRC1 complex, it mediates DNA double-stranded break repair and apoptosis evasion, and cooperates with PRC2 complexes to facilitate the epigenetic inhibition of the CDKN2A locus. And we know, of course, that BMI1 does function as a transcriptional repressor in this setting, and it really exerts oncogenic properties, particularly in CDKN2A deficient models of disease. So I just want to mention one more thing on this slide because it does come up a little bit later. In medulloblastoma, we also know there's a transcriptional regulatory feedback loop between MYC and the SWI-SWIFT remodeling complex that is one of the trithorax complexes I mentioned earlier. So SWI-SNF can actually evict the PRC1 and 2 complexes from the CDKN2A locus. And that indicates there's a very vital role for all of these epigenetic regulators in MYC-dependent cancers. One of the genes that really classifies group 3 and is known to occur specifically in group 3 medulloblastoma is MYC. MYC is thought to be a driver in group 3 medulloblastoma. And so we do think that these epigenetic regulators have a role in regulating MYC-dependent cancers. So we know that BMI1 is highly expressed in a plethora of very aggressive cancers, including colorectal cancer, breast cancer, prostate cancer, non-small cell lung cancer, and neuroblastoma, as well as DIPG and GBM. And so this has really become a key focus in our program because we think that this gene in cancer represents an excellent therapeutic target. So what you'll see here is some data from a paper that David recently published establishing this fact. And here we've already let you know that BMI1 expression correlates with poor clinical prognosis. But here what we do is we knock down BMI1, and here you can see, BMI1 protein levels in 2 very aggressive recurrent medulloblastoma lines. So these are those rare lines that are captured from patients who have already been treated and the cells have come back post-therapy. And these lines in particular, D458 and MB002 from Dr. Jay Cho, are highly show overexpression of BMI1, whereas even compared to the normal neural stem cell population, it's much higher in these recurrent medulloblastoma lines. And when we actually knock down BMI1, either with an shRNA approach or a CRISPR knockout approach, or in this case with a chemical approach, we have a small molecule inhibitor from our colleagues at PTC Therapeutics, which is a wonderful company based in New Jersey. And they are very interested in epigenetic regulators and building covalent small molecule inhibitors against them. So they have an excellent BMI1 inhibitor. This is the earliest version of that inhibitor called PTC-028. And we can see that when we inhibit BMI1 either chemically or genetically, we see a big decrease in both proliferation of these aggressive recurrent medulloblastoma lines and a very large decrease in self-renewal, as shown here. And in fact, we are now interested, since I mentioned to you that BMI1 is expressed in both cancer stem cell and normal stem cell populations. We don't have an EGFR variant 3 situation here. We don't have a tumor selective target. And so we really do have to think carefully about therapeutic windows and how we target with a BMI1 inhibitor, because you simply cannot treat a 5-year-old child with an actively developing brain and give them a drug that could target their normal neural stem cell population. I think you would just be recapitulating the same systemic toxicities against neurocognitive factors that we see already with radiation and chemotherapy. And so what we've done here is a rather elegant mixing experiment where you see the burgundy cells are medulloblastoma MB002 cells, and the gray cells are normal human neural stem cells, actually fetal human neural stem cells. We have these lines also in our lab. And so we mix these cells together, and then we treat these cells with varying concentrations and doses of the BMI1 inhibitor. And what we see when we do flow cytometry is when we see that there's actually a therapeutic window where it takes, the IC50 is 10 times higher in the normal neural stem cells compared to the medulloblastoma stem cells. And so that means that we can actually use a 10 times lower dose and kill only the medulloblastoma stem cells shown in burgundy and still spare the normal neural stem cells. So fortunately, we have this therapeutic window, and we believe that this is predicated very much on a gene dosage effect. It's simply the fact that I showed you on the prior slide that BMI1 is so much expressed at so much a higher level in medulloblastoma cells that forms the basis for a little bit of sparing of the normal neural stem cells. And so this is a very positive thing to find out about our inhibitor. When we target BMI1 in vivo and we're able to administer this drug, which now the new version, PTC596, which has already been in clinical trials in adult patients with solid tumors, has an excellent bioavailability, and you can even administer it by oral gavage to the mice. And so you can see here that when we target medulloblastoma MB002 engrafted in our mouse model, you can see that we have an excellent reduction of tumor burden in both the brain and also very fortunately in the spine, that leptomeningeal disease that is so hard to treat. And we also see a very good survival advantage in the mice. And so this actually has clinical relevance, because not only do you have a targeted therapy that so far in adult clinical trials has shown a very, very good toxicity profile, very low toxicity, and has excellent bioavailability and can be dosed orally. And then on top of that, you have BMI1 that can serve, BMI1 immunohistochemistry, for example, which can serve as a clinical biomarker, not only to select the appropriate patient population that may be a good candidate for this therapy, but also to follow progress of their therapy. For example, if you could recover cells from a patient's CSF and perform surveillance. So with that in mind, we seek to develop this program in children with both DIPG and recurrent medulloblastoma. And this is an example of a child who was treated at Memorial Sloan Kettering Hospital on compassionate release with the BMI1 inhibitor. And the way that we were able to determine that this child was a good candidate for a BMI1 inhibitor is this is a child with a group 4 medulloblastoma. She relapsed 2 years following completion of her standard of care therapy. And you can see here that this is a normal human brain stained for BMI1, very little of it except for in the subventricular zone. And here you can see that this is the patient's tumor at primary presentation, and then this is the patient's tumor at relapse. And this is a very rare patient where she actually went for surgery and was biopsied again at recurrence. And you can see that BMI1 is almost tripled in expression. And in fact, almost 90% of this child's recurrence expresses BMI1, meaning that this is probably a very good candidate for this therapy. And so now in the lab, we're exploring the use of the BMI1 inhibitor in our therapy-adapted mouse model. And we're trying to observe whether co-administering the BMI1 inhibitor along with standard of care therapy or delivering BMI1 inhibitors sequentially after standard of care would form the best therapeutic premise. And it's really about whether we think that, that administering the BMI1 inhibitor upfront will get rid of this population, or whether it's better to wait until standard of care therapy is completed for those cells to emerge and then to hit them with the BMI1 inhibitor. And what we see here, of course, is a prolongation of survival, whether you administer it sequentially or whether you administer it together with standard of care, but perhaps there's a slight advantage with the sequential delivery. So this brings me now to really speculate more on carrying our BMI1 program forward to really understand how the BMI1 inhibitor is working. We have seen that there's a very good effect of the BMI1 inhibitor in vivo, but you notice that it did not eradicate the relapse disease completely. Even after treatment with the BMI1 inhibitor, there is still persistent relapse. And so that brought us to believe, as does PTC Therapeutics, that perhaps it's best to combine the BMI1 inhibitor with another therapeutic to administer following standard of care when children relapse. And so in this case, we're able to really deploy our model systems of recurrence to really answer this question in a focused and targeted way. And rather than just doing sort of a guessing game of what should we combine the BMI1 inhibitor with, my lab sought to take a rational approach to trying to discover what is the best combinatorial therapy together with the BMI1 inhibitor. And so I'll just share with you a little bit of unpublished data where we've been using clonal tracking as well as functional genomics, specifically whole genome CRISPR screens, to identify new functional synergies that can work alongside of standard of care as well as BMI1 inhibition. So the first thing we did is cellular DNA barcoding. So in collaboration with Dr. Jason Moffat at the Donnelly Center at University of Toronto, we were able to perform or use his library, which is comprised of over 2 million unique DNA barcodes, drop that on our medulloblastoma cells, and then design a very elegant in vivo experiment to try to understand what are the clonal dynamics of these cells as they go through therapy, both standard of care and with the BMI1 inhibitor. So here you can see that we infect the cells with the barcode library, and then we put the animals through 4 arms. You have the standard of care alone, you have the control arm with no treatment, standard of care plus BMI1, and the BMI1 inhibitor alone. And we've been able to collect those samples post-therapy at endpoint and then try to understand the clonal dynamics that happen. So here you can see that we can successfully extract and identify those barcoded cells. There is the very expected big drop-off at engraftment that you see here, and this simply represents the in vivo clonal dynamics of roughly 10% of cells engrafting overall. And you can see here that we've been able to recover on average 10,000 barcodes per brain. And I'll just share with you, we haven't really, this is really quite fresh data, and so it hasn't been yet beautifully visually displayed, but I'll share with you the sort of clonal dynamics we so far uncovered. Barcoding is a very time-consuming and elegant experiment, but it really only answers one question. And the question it answers is your recurrence driven by one clone, or is it driven by multiple clones? Is it polyclonal or monoclonal? These are really the only kind of questions that we can answer. And so I just want to show you here that in the control mice, you can see these are the clones recovered from the brain and the spine. And you can see that in some cases, there are unique clones that drive spinal recurrence, unique clones that drive recurrence in the brain. And here are the clones that drive recurrence in both compartments, the brain and the spine. And you can see that, that in the control animals, the recurrence is definitely polyclonal. And although we have a therapeutic bottleneck, there's still multiple clones driving recurrence. Every dot here represents a single barcode. Then you can see that with standard of care, fortunately, as we would think, our current gold standard care further narrows the bottleneck and gives even fewer clones at recurrence. But very, very interestingly, whenever we did this, and this is actually representing 3 mice with the same result, when we treat with both standard of care and the BMI1 inhibitor, we can only find a single clone that is driving recurrence in both the spine and the brain compartments. And you will all be intrigued to know that it's the same clone that we're finding in different mice. And so this is very suggestive that the BMI1 inhibitor is a good addition to therapy, but there's still something driving recurrence. And we really want to understand what that is. And so that brings us to our next experiment, which is the genome-wide CRISPR screen. So we've been able to harness genome-wide CRISPR screening technology in our lab to identify sensitizers and resistors to BMI1 inhibition alone and in combination with standard of care, and also without the drugs, just to be able to understand what are the essential drivers of medulloblastoma recurrence. So two different types of CRISPR screens we've been doing in the lab. So here's the profile of the first type of CRISPR screen I mentioned, where we're essentially subjecting the same, now this is all an in vitro experiment, but otherwise the arms are the same as the prior barcoding experiment that I shared with you. And you can see that we have to grow 400, 500 million cells in order to accomplish this experiment. So this is a very time-consuming and costly experiment. So what I'll do is, while we were waiting for waiting the several months for the genome-wide CRISPR screen to be completed in MB002 with the BMI1 inhibitor, we wanted to do some sort of background work and try to understand what are the essential regulators of these cells, and also keeping in mind the comparative biology approach of trying to understand, are genes that are essential drivers in medulloblastoma also shared in normal neural stem cells, because those are not functional drivers we want to target. We want to know the tumor-specific functional drivers that may not be expressed in normal neural stem cell populations. So we went back with our functional genomics collaborators in the Moffitt lab, and we composed these Bayes factor scores from known databases. First of all, we have MB002, where we simply conducted a genome-wide CRISPR dropout screen, try to figure out which guides are actually driving medulloblastoma recurrence. So we were able to take a time point from this screen, as well as normal neural stem cell screens that were performed by our colleagues Toledo et al. And McLeod et al. From Peter Dirk's lab. We were able to compile all the data from these screens and normalize them and come up with 1,167 genes that were essential, it's shown here in red, only to medulloblastoma and not expressed in the normal neural stem cells. So while we were looking at these genes, we were also able to look at some other screens that were done in the Moffitt lab on the haploid cell line HAP1, which the Moffitt lab primarily uses to generate all kinds of interesting genetic interaction information, but also because they're very easy lines to screen. So what you can see here is we compared HAP1 cells that were treated with the BMI1 inhibitor PTC028 with some of the data that I shared with you on the prior slide. And so here in blue, you can see sensitizers to the BMI1 inhibitor that are essential in MB002. And in yellow, you can see the BMI1 inhibitor suppressors that are essential in MB002. And so we're compiling all of this data while we're waiting for the final results of our screen. But we were able to predict potential context-specific drug-gene interactions that may potentiate or sensitize the cells to the BMI1 inhibitor in MB002. And I'll direct you here to the top hits, GSG2. I think I'll have them better summarized on the next slide. But I want you to notice that along with our interesting gene SPAG5 that also comes up in our brain metastasis modeling, you can see that there's several genes here from the SWI/SNF trithorax gene chromatin remodeling complex that I mentioned earlier. So it's very interesting to see Jeremy Rich targeting EZH2 and BMI1 in GBM. And here we find similarly that there's other regulators in the polycomb complexes that are coming up on our hit list for drug interactions. So our top candidate genes that we put together all of this data while we were waiting for the final results from our formal screen, we put all of this together and realized that there are actually some very interesting candidate genes that are not essential in normal neural stem cells, but essential drivers of MB002 or recurrent medulloblastoma that could present possible co-therapies for the BMI1 inhibitor. And I'll come back to GSG2 because this is an absolutely fascinating gene that actually encodes the serine threonine kinase haspin. And here you can see when we perform an analysis on the data from our prior screens, this is called drug Z scoring. A positive score indicates a gene whose loss confers resistance to the BMI1 inhibitor, whereas the negative score indicates a gene whose loss increases sensitivity to the BMI1 inhibitor. And so what we suppose here is that the sensitizer genes represent rational targets for the development of synergistic polytherapies with the BMI1 inhibitor. And in fact, when we go back now and finally look at the results of the screen that we waited months for to corroborate, it's good to see that all of our other homework that we were doing paid off. Because when we look at the MB002 screen now treated with the BMI1 inhibitor, we find that the one hit that seems to be really pervasive is again GSG2. So we identified this not only now as an essential gene in MB002, but it's also come out as the most potent sensitizer to the BMI1 inhibitor, not only in HAP1 cells, but in our formal medulloblastoma screen as well. So this is very interesting. The serine threonine protein kinase HASPN actually phosphorylates histone H3, and it's known to interact with other regulators of medulloblastoma, including being involved in a positive feedback loop with Aurora B kinase. And we found that that this serine threonine protein kinase is actually directly phosphorylated by PLK1, and PLK1 is a known mediator of medulloblastoma genesis. So we went on to look at GSG2 knockout, and we found that it impairs both proliferation and self-renewal of MB002 cells in medulloblastoma. And so really now we're very excited about this to go on and look at whether GSG2 knockout increases the efficacy of the BMI1 inhibitor, both in vitro and in vivo, which is our next rational combinatorial experiment. And in fact, when we were able to find there's a known small molecule inhibitor of HASPN kinase, and when we got the small molecule inhibitor in the lab, we found that it actually has quite a reasonable IC50 and seems to be potent against MB002. So we'll go on now to combine this inhibitor with the BMI1 inhibitor and see if we have a more durable and tractable response, and whether we can eliminate that final clone that we saw in the barcoding data and perhaps block recurrence altogether. So this just brings us back to all of the sort of baseline homework we did has also revealed there are other tractable combinatorial pathways for targeting with the BMI1 inhibitor listed on this slide. And so we definitely have many backup options if our lead hit fails to materialize as a good combinatorial therapy. So I think I'll stop there. That's sort of the end of the BMI1 inhibitor story. I'm happy to take questions there before I go on to the next section.
Thank you, Sheila. So there's one question from Dr. Zhao. Is the BMI1 concentration different in the relapsed tumors, and does the treatment window of the inhibitor change in relapse cases?
Yes, that's an excellent question, Dr. Zhao. So we find that actually it's much harder to kill the relapsed medulloblastoma cells, and it requires the IC50 definitely goes up at relapse. And we find that primary cells are more sensitive, but they comprise a smaller compartment of the overall tumor, whereas the BMI1 cells tend to dominate at relapse. I think they're the dominant clone, and we definitely find that they are more resistant than in the primary stage. And we don't know if that's related to a gene dosage effect as well.
All right, so no other questions so far. So why don't you continue, Sheila?
Great, I will. Probably what I'm going to do, because I'm speaking far too long and I've already, I'm going to skip this story and I'm going to move on to cover one more topic in my last 15 minutes. And I can certainly speak more at the end. So I'll probably land here and just tell you a little bit more about the applications and the utilization of the model that we developed. So my student, David Bakshinyan, worked very closely with another master's student in the lab, Ashley. And Ashley was observing, all of the work that David was doing with the genomic, transcriptomic, and proteomic characterization of this model. And she realized that the stage that she was most interested in studying in her master's was the spinal metastasis in particular. We had this nice benefit of being able to compartmentalize the recurrence and study them separately. And she was particularly interested in trying to figure out noticing that we had a differential gene expression profile between the brain-driven metastases and the spine-driven metastases. And she wanted to figure out whether there were specific ways to target the spinal compartment. And so you can see what she did here is she actually went on to perform, to recover the cells from the relapse of the MB002 treated mice. And of course, we have the cells from all of the stages of this model frozen down and to be used for other purposes. So Ashley focused specifically on the relapsed cells, and in collaboration with our human stem cell screening facility, we actually performed an initial chemical screen of 640 kinase inhibitors in collaboration with the Ontario Institute for Cancer Research. And this high throughput screen actually identified kinase inhibitors that very specifically targeted the group 3 gamma version of these spinal metastases. And we found 47 compounds that reduced the cell viability of this very aggressive tumor by over 50%. And we went on to screen from those 640 down to 47, and we found 8 compounds that had good blood-brain barrier permeability for the purpose of in vivo studies, and 5 that we eventually settled on that had very good PK/PD. And so you can see these are the 5 that we found, and notice that there is a PLK inhibitor, a bromodomain constraining protein up here on the top that appeared on our list of hits. We were able to identify these inhibitors and then went on to ask our key question of whether these inhibitors could specifically target medulloblastoma and still possibly spare normal neural stem cells. And here you can see that of those 5 compounds, we found 2 that seem to have a lesser effect on the normal human neural stem cells and more of an effect on the medulloblastoma stem cells. And those ended up being the CHEK1 and the PDGFR beta inhibitors. So now we went on to perform some blood-brain barrier permeability assays to see if these would be viable options to take in vivo, and did some in vitro stem cell assays and found that both of these compounds actually do significantly reduce the stem cell properties of MB002, both self-renewal and proliferation. And so at this point, we're looking at taking these compounds in vivo specifically to see and to use our imaging facility to monitor and see whether we actually block or prevent the dissemination of the cells going down the spine. And so we'll be pre-treating these mice with these 2 compounds in separate trials to try to determine whether we can actually block the spinal metastasis. And so, of course, the aim here would be to see, is there a therapy in the future that we could co-administer in children who we think are at particularly high risk for spinal dissemination of disease? Could we somehow prevent that from happening in the first place? Would it be possible to somehow give an anticipatory targeted therapy that could block the cells that are predisposed to move down the spine and to somehow add this to our current standard of care to perhaps improve the rate of spinal dissemination in the future? So just to show you how in the development of this model, we've been able to refine the model and move on to study other important components of this deadly disease. So I think I'd like to finish in my last few minutes on one last story that we have that also is a different way that we took about targeting self-renewal in medulloblastoma stem cells. And this story is from my MD PhD student, Branavan Manaranjan, who following his graduation has started his neurosurgery residency at the University of Calgary in Alberta. And so Branavan came into his PhD with a very interesting question. We basically, as he was learning about medulloblastoma, noticed the fact that the Wnt subgroup has a very, very good outcome compared to the other medulloblastoma subtypes. And the Wnt subgroup in particular has a 5-year survivorship of over 90%. It's such a relatively good medulloblastoma to have that there are clinical trials going on as we speak looking to de-escalate our current radiochemotherapy. And so the question that Branavan asked is, how would it be possible to recapitulate the phenotype of the Wnt medulloblastomas by activating Wnt in the poor outcome medulloblastomas? And so in that question, this is actually a lesson that we learned from melanoma. And I just want to share with you an interesting story. At the beginning of my tenure here at the McMaster Stem Cell and Cancer Research Institute, one member of our scientific advisory board was actually Randy Moon. And we had the great fortune of working with Randy, who of course is a world-renowned Wnt signaling expert. And on his tour of the facility, he came to stick his head into my office. And he just basically said, this was before the publication of this paper that I'm sharing with you here, he said, Sheila, I really think you should look at Wnt activation in medulloblastoma. And then he left. And so really when Randy Moon, signals to you that you should be studying something, it's a good idea to follow his advice. So you can see here from melanoma that Dr. Moon had already shown that activated Wnt and beta-catenin signaling medulloblastoma is actually a positive factor in reducing tumorigenicity. And in fact, several publications have come out and shown the same phenomenon as opposed to colon cancer and GBM, where Wnt activation is known to be a driver of aggressive biological behavior. It seems to be the opposite in melanoma. And in fact, if you look through pediatric medulloblastoma, we know very well that activated Wnt signaling in many different studies has been shown to be a positive prognostic factor and promotes survival overall in pediatric medulloblastoma. And so we went into the lab, and we were fortunate enough to have operated on some children with Wnt in group 3 and group 4 medulloblastomas. And so we had patient-derived lines from our own patient population from those subgroups. And here you can see that when we profiled the Wnt subgroup of tumors, we found that key self-renewal, master regulatory self-renewal stem cell genes, such as SOX2 and BMI1, were expressed at very low protein levels in the Wnt subtype compared to group 3 and group 4, where they were very high. And you can see, of course, as I've shared with you already, BMI1 is a predictor of poor prognosis, and in fact, SOX2 is as well. And so we went back to the group 3 and group 4 medulloblastomas after a paper was published suggesting that although medulloblastoma patients have a dominant molecular subtype, they may actually have cells of all different subtypes comprised in their single medulloblastoma. And so we went back, and using a top flash reporter system, we went to profile our group 3 and group 4 medulloblastoma cells to try to figure out if there were any endogenously Wnt activated cells within the tumor. And in fact, we found that when we did some single cell profiling, that 8 to 10% of cells within our group 3 and group 4 medulloblastomas actually show a high Wnt signaling signature, and also in our assays had high top flash activity. And in fact, what you can see is that when you sort those cells that are here we call TGP positive and negative, you can see that the cells that are Wnt activated, the TGP positive cells, actually form very benign looking, superficial, non-invasive tumors compared to the TGP negative cells from the same group 3 medulloblastoma, which form very invasive, aggressive medulloblastomas. So here you have cells from the same patient's medulloblastoma. One population of cells is indolent and doesn't form tumors very well, and those mice tend to survive for longer, whereas the cells that, the other population of cells is the opposite. And the one factor that distinguishes these cells is the benign and indolent cells had endogenous Wnt activation. So we found this very, very intriguing. And we went on to use 5 different techniques to activate Wnt in otherwise Wnt unactivated group 3, group 4 medulloblastoma cells to see if we could convert them from their more aggressive phenotype to a much less aggressive phenotype. And here you can see that when we Wnt activate a medulloblastoma, whether that is genetically by beta-catenin overexpression, or whether a lentiviral transduction of a beta-catenin overexpressing construct, or whether it's by adding Wnt5a conditioned media to the cells, or whether it's by using a GSK3 inhibitor, which is another chemical way of activating Wnt, you can see that self-renewal decreases greatly in the tumors that are Wnt activated, and tumorigenicity decreases greatly as well. And here you can see this is the ectopic expression of beta-catenin in our recurrent medulloblastoma lines, reduces the overall tumor burden and greatly increases survival in vivo. So the final step that Branavan undertook is that we found this wonderful paper published in Science Signaling in a mouse model of Alzheimer's disease, where they used intranasal administration of a GSK3 inhibitor in this mouse model. And we went and bought that same GSK3 inhibitor, and we administered it to our mice intranasally. And we found that this greatly reduced tumor burden once again. So the mice treated with this L807 compound had a huge reduction in tumor burden in an otherwise highly aggressive recurrent group 3 medulloblastoma. So we were able to ascertain that Wnt activation was a really tractable strategy for reducing the aggressiveness of group 3 medulloblastoma, which may be driven by cancer stem cell self-renewal. So that just gave us another pathway to examine self-renewal. And unfortunately, I don't have time to share Michelle's wonderful story. This is my neurosurgery resident, Michelle Canetta-Smith, and she's presented at many conferences lately, her beautiful work examining Musashi 1 in our medulloblastoma models. But what I'll do is I'll simply finish to share with you the fact that in my lab, we've tried to use our beautiful models of recurrent medulloblastoma specifically to interrogate the molecular mechanisms and underpinnings of self-renewal in these cells. And we try to find different ways and different manners of targeting self-renewal. And whether we target self-renewal with a small molecule inhibitor against BMI1, or perhaps by activating Wnt signaling to reduce the expression of key self-renewal genes such as BMI1, or perhaps through post-translational regulation of genes that we uncovered through some elegant E-clip experiments that Michelle did in collaboration with the Gene Yao lab to discover how Musashi binds to certain transcripts and regulates medulloblastoma. Perhaps through all of these different mechanisms of targeting self-renewal, we can get closer to trying to develop new targeted therapies for children with recurrent disease. And I think I'll end there to make sure that I'm on time. And just to let you know that I've had a great advantage of working in Canada in the field of stem cell biology. We've actually had many great discoveries made in the field of stem cell research. And there you can see Till and McCulloch. Dr. Till was still working at Princess Margaret Hospital when I completed my PhD studies. And I had the great honor of meeting him and talking to him about our work. Of course, he went on to mentor Norman Iskov and Connie Eves, who I've been fortunate enough to work with, as well as Sam Weiss, who discovered normal neural stem cells in the mammalian subventricular zone, as well as John Dick, who sat on my PhD committee, right down to Peter Dirks, who was my PhD supervisor. So I've been very lucky to work within this very rich and highly accomplished stem cell biology community. And I think the Globe and Mail captured it very well when they published this article saying, if Canada's game is hockey, then our science is cells. So I will finish there. And thanks, Stephen, very much for the opportunity to speak to you today. I'm happy to take any questions.
Thank you, Sheila, for a fantastic talk. And before going to the Q&A session, since it's at the top of the hour, I would like to thank all participants for attending today's lecture. And I hope that we see you again next Thursday when Aviv Regev from Genentech will talk about single-cell atlases as roadmaps in pediatric cancer. So yes, Sheila, so there was one question which came from Dr. Schwing for your first part, coming back to the clones you found. Are these in the end patient-specific, or are these the same clones which come up in different tumors?
That's an excellent question. So far we've done the barcoding experiment, which you can imagine is very onerous, only with 2 medulloblastoma patient-derived lines. And we do find that the clones that come up of recurrence differ from patient to patient. Unfortunately, not mouse to mouse engrafted with the same patient tumor, but patient to patient, we do see different specific barcodes come up at recurrence. However, the pattern of clonal recurrence that you see is the same between patients. So that same bottleneck, which seems to be further enhanced by the BMI1 inhibitor, seems to be the same pattern among different patients, although not the same barcodes.
There's one question from Dr. Sung. His question, it relates to MYC. He's wondering if you would block MYC, is that sufficient to kill the cancer?
Well, that's such an excellent question. I think it's a bit of the holy grail of group 3 medulloblastoma. Is certainly, inhibiting MYC would be something that would be desirable, and we all know that that's an incredibly difficult thing to do. I think the quest for good MYC inhibitors has been long and not so fruitful, and unfortunately there's a lot of escape. And so I think we're thinking about trying to target genes that are perhaps upstream of MYC, regulating MYC. And in fact, that's why, I sort of brought up the fact that BMI1, the polycomb complexes, which seem to be a little more targetable, as demonstrated by their work today, they do have some regulatory effect on MYC, as well as the Musashi pathway, for example. So we're trying to find almost like ways of getting at MYC without having to directly inhibit MYC.
Great. I had, one question. Of course, some might wonder why I invited you, but you didn't talk about some of your cell therapy work you're doing. And I was just wondering, as you kind of see these small molecule inhibitors developing, what about combinatorial therapies? Are you kind of, as you set up your models and look at these inhibitors, for example, also try to look what these do to, the immune cell landscape, of these tumors?
Yeah, Stephen, that's an excellent question. So I think it's highly relevant and important to look at the tumor immune microenvironment as well as the microenvironment of medulloblastoma. And that will require really, really good syngeneic models, which we have and which you have, of course, at St. Jude's. Syngeneic modeling is essential there because, of course, in our PDX models, one of the big downfalls is the fact that our mice have a very, very sparse, almost non-existent tumor immune microenvironment. And so we can't properly recapitulate that in the patient-derived xenograft models. And so I think with excellent syngeneic modeling, this should be entirely possible to do and very important, because if I could take some lessons from our GBM work, I think with the GBM program, we realized that we need to design therapies that co-target the tumor and the time. Targeting the tumor alone, the cell intrinsic pathways, is not enough. So I do hope there'll be more research in that area. And I do foresee that combinations of immunotherapy with small molecule inhibitors could perhaps down the road maybe replace our standard of care altogether.
Great. There's one question from Dr. Yao now, and it is about, given the heterogeneity of group 3, group 4 medulloblastoma, have you observed any subclones that increase in aggressiveness when you activate Wnt signaling?
That's an excellent question. So in terms of activating Wnt, what we see is that so about 8 to 10% of the tumors that we've surveyed, group 3 and group 4 tumors we've surveyed, seem to have an endogenous Wnt activation anyways. So what we're looking at is trying to turn on Wnt in the remaining 90% of cells. And what we find is that we haven't seen any adverse effects of cells escaping the Wnt activation and becoming even more aggressive. Not at all. In fact, the Wnt activation in these cells does seem to calm them down and make them indolent. And interestingly, it's not that the tumor burden is eradicated. In fact, there is still tumor burden, but it's almost like, if I could compare this to converting a GBM to a low-grade glioma. That's the effect of Wnt activating, if you will, the group 3, group 4 medulloblastomas. There is still tumor, but it's now behaving as an indolent chronic tumor that could be perhaps managed over time. So I see the Wnt activation strategy as being more of a trying to turn a very aggressive grade 4, WHO grade 4 cancer into a chronic disease. And if we could make those tumors into something akin to a low-grade glioma, the child could survive with that. As a neurosurgeon, I manage many children with low-grade gliomas over time. They have excellent quality of life. They have great survivorship. So I think that's more the focus of that program.
There's one question coming back to drug screening from Dr. Blaszczuk about differences between, doing assays on a single cell or in a 2D versus neurospheres assay. So if you could comment on that.
Yeah, that's another excellent question, because it comes back to the effect that's another limitation of our in vitro assays, where we have single cells in a dish or even single spheres. Are we truly recapitulating a drug effect that we would see if there were a proper tumor immune microenvironment? So I'd like to point out that there are many labs that are now developing very beautiful organoid model systems that recapitulate not only the brain microenvironment, but also the tumor immune microenvironment. And there's one example I can give is Dr. Howard Fine, who's developed a glyco model, the glioma cerebral organoid model, that comprises not only GBM cells in a dish, but also brain microenvironment cells, astrocytes, neurons, microglia, and other immune cells as well. So I think these models are actually ideal to optimize for these drug screens. And it's a question of how can we adapt them for a high throughput drug screen where you have, your 384 well plates. Can you possibly, put organoids into those instead of spheres? So I think that's a great point. Right now, we've only assayed the effect just on the cancer stem cell population alone, and we don't have an idea of drug responses with the proper microenvironment.
Great. So right now there are no additional questions. So Sheila, I would really like to thank you for a very stimulating talk. And as I said before, I wish you could have invited you in person, but we'll do that once COVID travel restrictions are limited. Thank you so much for taking time to give us the talk today.
Thank you for inviting me, Stephen. I look forward to meeting you all in person soon.
Bye. And I would also like to thank all participants who stayed on for the extended period. Thank you so much. Bye-bye.
Thank you. Bye, everybody.